going to start to talk about sequence to function models. And the idea here, the animations we're working, is that you're taking DNA, which you can see a little part of on the left, and you're going to build a model that predicts a functional genomic output, like some of these genomic tracks that we see on the right. And oh, I guess they're working. Usually the model is some kind of neural network, and these things have gone amazingly good. So this is a picture from Informer, and one of these, I think the top is the experiment, and the bottom is a prediction on some genomic sequence that the model has never seen. So they're highly similar. So we have these models, and we can kind of do a certain approximate what the cell is doing, how it's interpreting its genome. So what are these models actually good for? I mean, it was kind of a fun exercise. Can we even do this? Is it possible? It's definitely possible. So what do we do now? So one thing that these models get us is the ability to predict functional output on arbitrary sequence, like sequence that we've never seen. We can do variant interpretation for population genetics. We can actually design completely new sequences that have good properties. But the model actually is somehow recapitulating cellular processes. So it means at some level, it must understand what is the cell is doing. And so we can ask kind of to try to extract some biological insight from the model. So ideal case, this is something I would be able to do, but in the future, I hope to be able to do, is take one of these models and output something that looks like this, which is a textbook picture of immunology, like lineage differentiation, with all the factors that control it, right? Something that's been worked out through biochemical assays. So we can't quite do that yet, but we can already actually get some insights from the models that we have. So if our model is a black box model, it might seem like there's not much we can learn, but that's actually not true. We can learn things even from the black box model. So we can ask like very simple questions. What is the right input set towards like some kind of biochemical activity? How much of the DNA do we actually need to predict this output? And so for gene expression, it turns out, this is quite a lot. And that is like one of the selling points of the Informer gene expression model, where they use quite a much larger window size than previous models, and we're able to get better performance. So from this, we can conclude like, well, if I have to use 1 million base pairs, and that is better than 100,000 base pairs, then there's some biochemical process that operates at this larger scale. So we already knew that, but that is something that's actually kind of made tangible through the performance. So kind of what's something that we don't quite get is the why and the how. And this has actually kind of been reflected in all sorts of, the Informer model is so popular that there are papers written just analyzing it as a scientific object in its own right, which I think is kind of interesting. So, for example, one paper here that's referenced on the bottom looked at like, okay, so long range sequence is important, but how much information does it actually contribute? And they did a very simple thing where they just like kind of zero out the tail ends of the input sequence. It turned out if you do this, the variance explained by the long range is actually quite small. So yes, it was a computational like achievement, but in the previous slide, it was not actually an apples to apples comparison. They compared it to a different model. So in an apple to apple comparison, it seems that long range is not doing a whole lot here. And actually, this is not biological. That was the other conclusion of this paper, where if you look at how much regulatory contribution is actually made by enhancers at different TSS distances, so you can look, there's the observed contribution. So it does kind of trail off depending on which data set you look at a little bit, but it's actually, you can have pretty strong contributions at large distances. For the Informer model, it really drops. So this kind of underscores that, while you can learn certain things from these black box models, it can be misleading, and you certainly don't get to learn everything. So for our work, we took inspiration from this deep learning paper, which is titled Deep Learning Immune Cell Differentiation, but it's really trying to leverage the potential for interpretation, the potential for learning biology. So this is AI-TAC model, which is a model, but also an interpretation framework. So they have kind of a fairly simple architecture that the team, it was optimized for its interpretability, what they could do with it post hoc rather than just absolute performance. And the prediction task they're looking at is chromatin accessibility across cell types, and they were using correlation as the objective. So really trying to understand differences across cell types as opposed to the absolute amount. And they had also a scientific objective, which is to understand the immune cell differentiation. So how do they actually do this? So this is, sorry, a little bit. So in the first layer, where we have a large convolutional network, on the first layer, we can interpret it as TF binding motifs, which you actually can represent the first layer in these very familiar PWM kind of logos. And you can track kind of how these motifs influence the final layer activation, which is activity across cell types. And this gives you a PWM to cell type activity map. The PWM sometimes can be, these kind of filters, they can often be mapped to transcription factors, in which case they're given a name on this plot. Sometimes they're not identified, and then they're just given a number. But from this map, we can actually tell kind of that we're getting correct things. For example, we have PAX5 as regulating the B cell lineage, which is known. This is a nice system because a lot is known. It's immunology, been working on it for a long time. Or ROR gamma for ILCs, and a lot of many other things in here are also correct, which kind of gives us confidence any new things we find might also be correct. Okay, so we have this basic framework. Okay, first layer is interpretable as these nice kernels. Here's a mapping between what the model learned and what we can actually find in a database. And you often see these kind of plots when people get good matches, they like to advertise it. What we get. So, but we had all these other layers in the network, right? The hidden layers. So what do? So I guess kind of a fun answer that people like to give is that like, oh, it's modeling, interactions and complicated transcription factor grammar. And we do have these really nice, well-characterized examples where you only get activity when the transcription factor binding sites are arranged in a very specific way. Okay, but they can also actually do kind of maybe boring things, right? So for example, they can refine the initial layer motifs. They can do kind of just very basic nonlinear calibration for like the binding probability of transcription factors, or for like nonlinearity in your assay output, right? Like chromatin can only be so open. Once it's open, it's open. And even if we are actually capturing interactions, we actually don't really know what kind of interactions we're getting, because we can have a linear effect, which is no interaction. We can have a synergistic effect, which is the kind of that's the kind of thing we get very excited about. But actually, an OR function is also nonlinear, right? You need nonlinearity to capture OR. And it's actually pretty hard to get this information out of the network. Okay, so I want you to kind of give a little bit of an overview of the data set we're working with, because it has some kind of specific characteristics. So we have 81 mouse immune cell types, and they're actually arranged in this nice developmental tree. So we have kind of parent-child relationships. And because this is an immunology data set, we have these nice textbook pictures for some of these transitions. We actually know how they're regulated. There's positive regulators, negative regulators. And for specific transitions, we actually have a lot of information. So the other observation about this data set is that there are 7 main lineages. And when we do predictions, the models perform much, much better at doing across lineage than within a specific lineage. So I've highlighted there like a particular subset of alpha beta T cells, and which of course come in all of these different varieties, like follicular T cells. We have regulatory T cells, so that's all kind of within that lineage. They're all very similar. On the gene expression level, on the attack-seq level, they're similar. They're functionally very different. So we do want to really understand where that function comes from. And these models that are trained in this way really struggle to get these quantitative differences right. So for most of our study, we're just going to be working at the lineage level, because that's the one thing that we really understand. So we can, sanity check our results. And well, the models aren't working so well on the within lineage predictions. So, one thing we asked is like, well, how about if we just build a really, really simple model and try to understand what is actually the performance gap between our model and like a fully black box model that is optimized for this data set. So this is our attempt to build a really simple model. So we're doing elastic net regression on the maximum motif score for all cis BP PWMs. Right? And you can see the results there on the plot, and the results are R-squared. So one thing you might ask is like, this R-squared seems kind of low. You should really be getting like 0.5, 0.6, something like in that range. So the reason for this is this is for the Z-score for predicting differences across cell types. And that is a harder problem. So if you translate it back onto the absolute scale, you do get like kind of the more familiar values. So, okay, we definitely have a performance gap, like no surprise there. But the linear model does have some advantages. So for example, this is a totally convex problem that has absolutely no training dynamics that I have to worry about. I will get exactly the same answer every single time that I run it with like really nice sparsity. It's actually very easy to select hyperparameters. The output is actually immediately interpretable as a TF to cell type map. And but you know, there's also some kind of, obviously it does not really that performant, so that's one problem. And you know, we did have to do some kind of feature engineering. It's hidden here on the slide, right? I said we took max motif score. Why do we do that? I mean, people do other things, right? Sometimes people like draw a threshold and they count the motifs. Why didn't we do that? We had to make this decision. We picked this one. Definitely max is better than average, I'll tell you that. But is it better than counting? Well, it depends on your threshold and it depends on the motif. So, right, we had to make some choices and we had to like give up some ability to optimize things. So we wanted to kind of bridge all of this and create something that had, you know, solved at least some of these problems. And so we want to increase the capacity of the linear model and improve performance, but we kind of want to do it in a way that maintains interpretability. And so the main idea is we're going to build like a kind of multi-layer deep learning type model, but it's going to have no uninterpretable hidden layers. So every layer just references transcription factor activation, and layer construction is biologically inspired to the extent that we can, you know, handle it. And we call this model the totally interpretable sequence to function model, or TISFM. This is the architecture of the model, and I'll kind of step through it one little bit at a time. Okay, is there a clock in here? Because my watch like ran out of battery. That one doesn't work also. All right, well then. Hopefully I'll
give you like the 10-minute one.
Okay, fine. So all right, so this is the architecture. So we start out kind of like any other neural network sequence to function model, where we have a convolutional layer kernels, and they're actually the one thing that we do differently, but actually people do this quite a bit in the literature if you really read the method section, is you initialize it with known motifs. And so we freeze these parameters at first, and then we can actually optionally unfreeze them later and get them, let them be optimized as well. So our input is just one hot DNA embedding. We have a we also have a positional weighting, which basically gives the weight, because one of the questions that you might ask is like, well, what is the relevant region for predicting the signal? And people have like settled on certain values, but maybe they're not perfect, and it's kind of an annoying hyperparameter. So this way you can automate this learning, because the positional the weights can just trail off at the end and just mask everything that is not relevant, so that way it's automatically learned. We have this kind of it's called an attentional layer, but basically it just kind of turns one hot into numbers to give the model a little more capacity. Well, it turns out that didn't matter so much in the end, with flexible motifs there also optimized. And so taking these two things, we just do a standard convolution, basically the same way you would do in any model. Now, after the convolution, what we do is we apply a per channel nonlinearity, which is basically the one that we picked as a trainable sigmoid activation. So the motivation for this, we have these kernel scores, which is basically like a log odds. And That's actually not, it's not really biologically interpretable quantity because it's not, doesn't actually tell you like what the binding probability is. Ideally, like we would like output a number that is like probability that site is occupied. So certainly you need some kind of nonlinearity here. And so the way we decided to model this is we use this nice like sigmoid function, which is in biology shows up a lot. Not so popular for building really performant neural networks, actually. And so it just has two trainable parameters. So we have like a separate calibration curve for each motif. So there's there's an offset and a slope that you can learn. And so you can get all of these kinds of transfer curves that transfer your continuous values onto the 0-1 scale. So we were really hoping that we would learn like probability of occupancy. We didn't, but anyway, we tried. So and the next piece of the model is this trainable pooling layer. So remember how I talked about like how do I aggregate over a bunch of motifs? I can take the maximum value, I can do counts, I can do an average value. That tends to not work so well, or at least not until you kind of calibrate the scores. So we actually use a trainable pooling that just basically smoothly interpolates between max and average. And this is done for every transcription factor separately. And we had actually a specific hypothesis about what we would expect to see. So transcription factors have really wide ranges in how much information content there is in their PWMs. And so there are certain transcription factors where I see one of the sites, like I can be pretty sure that something is bound there. I mean, this is all kind of relative. And there are some transcription factors that have very degenerate sites. And there's lots and lots of them over the genome. Like I just like, in terms of how many proteins there are and how many sites there are, they can't possibly all be occupied, right? So for those sites, we're definitely sure that like there can't be 100% occupied. And we think that like maybe in this case, it might be in the case where we have like really high specificity sites, it might be the case that we really just want to take the max, that adding another high specificity site doesn't do anything. But in these cases where we have low specificity sites, maybe it does actually make sense to do some kind of average. So that's the hypothesis. We'll see if that works out. And finally, we have this module that we call the interaction layer. And just quickly explain how it works. So basically the interaction layer, so you have these motif scores, and now just I have one score for the whole sequence because I've maxed or average pooled it or something in between. So now it's just pooled. This is just now has motifs. And so I learned this interaction matrix, and the output of this is basically just a number between 0, 1 that gets to tune up or down with 0.5 being completely neutral, right? If everything is 0 here, you have no interactions, you get a value of 0.5. You get to tune up or down the activation for that transcription factor based on what else is present, right? So you can actually capture like a synergistic effect or a saturation effect. Your contribution can actually go down based on what else is around. Okay, and finally we get here, we get to the output. So if you think about it, we've maintained like the motif dimension is always the same and it always has names. Throughout this whole process, I actually can assign a specific biological entity to one of these features. Right, so I'm always working on the motif layer. It's been transformed, but you know, the dimensionality fidelity is preserved. And so from this final motif representation, I just have like a basic straight up linear model, the way I had in my very naive elastic net implementation that's going to predict open chromatin activity across cell types. And as before, the final layer is actually still immediately plottable. It has names. So just kind of looking at some of the performance contributions for this model. So the way we express performance is different from the base like standard CNN AI tags, so which is why they can be negative. So we have some replicates here, we have error bars. So basically everything that we did does improve. Anything you take out, the performance will be a little bit worse. And the biggest difference was made by the interaction layer. And this is basically the same plot now, but when we unfreeze the kernels. So we were a little bit worried about unfreezing the kernels because they can really hypothetically at least change a lot. And in which case you will lose your ability to name them automatically. You will have to remap it back to the database. And you might fail to map some of them, right? Sometimes we have like filter 207. I don't want any of that. I want all of them to have names. Turned out that was not a problem. And actually, most of them were like just completely preserved. They matched back to their starting point. Like the best match was where you started. But this made the model outperform AI tag basically in all different configurations, and still the best model overall was the base model plus the interaction layer. Okay. So what does this look like? So as I said, we hope that the final layer is interpretable. Certainly we can plot it like this, right? It is named cell type by transcription factor. And actually we got very similar results to AI tech, right? So here's PAX5, we got it for B cells. We have for gamma for ILCs and a lot of other things on here. Makes sense if you're in immunology. Okay. So, okay, what about this pooling parameter, right? We actually had a specific hypothesis about what this should capture. And so we can actually correlate the pooling parameter with the information content of the final like optimized kernels. And this is what this looks like. And it's basically, not perfect, but very much showing the trend that we predicted, that the more information content you have, the more you prefer max pooling overall, right? So the lower information content filters, they really they kind of have a homogeneous synergy where you do want to look at multiple sites, multiple weak sites, and add the contributions up. Okay. So next we look at this interaction layer. And so this is just kind of aggregating over the total interaction contribution. And so the one thing we wanted to see is how the interaction, we have a lot of things that are contributing in the interaction space, and then we have transcription factors that are contributing to cell type specificity. And we wanted to see like if there are any patterns that emerge by contrasting these two. And so we have just kind of like an overall cell type specificity on the x-axis and an overall contribution to interactions. And the thing to note is, yes, they're actually quite different. And in particular, one thing that we note about the things that are kind of highly interactive is they tend to be not 100%, but they tend to be transcription factors that if you look at their expression, they are not particularly specific, possibly not at all specific, with CTCF being like the most interacting transcription factor. That is a housekeeping gene that all cells have. Okay. And so there's a general trend in where the different transcription factors would appear. And kind of zooming into the actual interaction matrix, so that was kind of the marginal over this whole matrix. But now we can actually look at the whole matrix, like focusing on some of these transcription factors that were highly interacting overall. And this is actually because we have kind of explicitly parameterized everything in terms of synergy and saturation. We can plot this, and the colors roughly correspond, and try to keep the color consistent. And so what we found is actually that the whole thing is very dominated by saturation effects. And at first I thought this was like extremely disappointing, because of course we want to see synergy, and we don't see it very much. But actually, this is quite consistent with like even basic things we understand. So, for example, CTCF is has a lot of saturation effects, and CTCF really just one CTCF site will just open chromatin. Like people have now done these assays where you can introduce a site onto like a methylated piece of DNA, and it will get like unmethylated and just be open chromatin. So it's a very strong activator of open chromatin, and really you just need one. And once you have one of those, nothing else matters, and you can put whatever else you want. It's not going to become more open. So that's actually fairly consistent with what we understand. So, right. So one of the other kind of really nice things we had in our linear models, we used elastic net, which means that we got a sparse model in our transcription factor to cell type map. So and when you're kind of building out these neural networks and solving them with some kind of SGD type algorithm, getting good sparsity patterns is, it's actually an ongoing research question how you do this. And we took an approach that was fairly recently developed. So that's the paper adaptive proximal gradient methods for structured neural networks. It's designed to work specifically with the atom optimizer and take all of the atom parameters into account. And so this is just a figure from their paper about how much better they're doing in terms of sparsity map patterns, like recapitulating correct sparsity patterns than the previous approach to prox SGD. So we just took their approach. Is basically like what it is just a different optimizer. If you look at it internally, it is not that complicated. There's a couple lines that are different from just atom, but it's just a slightly different optimizer, and it has a sparsity hyperparameter that you can tune. So the basic parameter actually it implements, you can do L1 and this the non-convex MCP penalty. In the paper it said MCP works better. I mean, the whole thing's already not convex. Might as well use the non-convex penalty. Doesn't really hurt the problem. So we also use MCP, and you can kind of get these, this is not really a path algorithm, but you can get like a path algorithm lookalike plots by turning up your penalty parameter. You can watch things drop out. And one thing about the MCP, the non-convex penalty, is like things drop out very suddenly, as opposed to kind of like falling towards zero with L1. They just, right? So I can look at one of these plots and tell you if you use L1 or MCP, they will look very different. So it has these kind of really nice properties where you get really aggressive sparsity and like very fast dropout of the parameters. And actually you can set this with cross validation. So there's some sort of like for this cell type, there's some optimal point, which is right there, and it's also here. And you can see like, so all of this black region is basically something that went to zero. You still have a lot of transcription factors left. We're not down to 5 transcription factors. So we still have a lot of like sequence modeling complexity. But then if you look at, if you keep going past the point of where you're doing the best and you just keep driving the model sparser and sparser, what is left are like your well-known regulators of that lineage, which is actually kind of satisfying. And yeah, and this is just a plot of like what are the final, like if we take the best MSE, how many transcription factors are we left with? And there's some kind of interesting patterns in here. I think, and this is kind of alluded to in the original AI tag paper too, that the T cells are actually kind of their model is more complex. It doesn't really have a super sparse description of the T cell, like a lot of like the B cells, we know what the drivers are and we get them, or the ILCs also we know what the main drivers are and we also get them in the sparse representation. T cells, it's hard to get a sparse representation, and that was also true in the original paper. And they hypothesized that like T cells maybe actually a kind of a default state. So and then we have like all these transcription factors that can drive you away from that state. And so I think, it's an interesting kind of observation that you can actually have different levels of complexity potentially for different regulatory programs. All right, so this is all the lineages. I told you I had 81 cell types, and we do. Yeah, and so what about what happened to all of this? So, well, we have this one observation that models, deep learning models really struggle with these subtle differences when they're trained on to predict these multi-output values. And this is actually, a really persistent problem in the sequence to function community. Lots of people have given lots of talks about this. Probably you've seen one of them. This is a nice paper that just came out maybe like a month or two ago, I would say, where they really did a lot of careful work trying to analyze what's going on and how to improve it. But the basic observation is just kind of highlighted in this plot from the paper, where if you have a cell type specific peak, which is there in the box, right? These are cell types. We have actual measurements, predictions, and where I have these common peaks on the plot, and then there's a cell type specific plot. The model fails to predict it. This is not something you really, unless you design your performance metric to really look at this, it's not something that's going to be immediately apparent, because there's actually there's a lot of similarity in the overall pattern. So overall, you're doing well, right, for that cell type. And this I found is like really fascinating. So if anybody has any like interesting perspectives on this, I would love to hear it. So okay, so it's like how do we deal with this? So it appears to be that this is a problem with really doing things in a multitask setting. And so one way is you can just not do multitask. So this was one of the suggestions by the authors. It's like, just train one cell type at a time. You will actually get better specificity. That's the light colors here versus the dark blue. Not 100% consistent, but overall that is a trend. And like, well, if I can train a bunch of single output models, shouldn't I be able to just like stack them and get a multi-output model that's completely equivalent? And for some reason, that turned out to not be right, because they actually, one of the things they're showing is a multitask with 8 times the parameters. So in fact, they multiply parameter space by more than the cell types that they had, right? So it should definitely be able to encompass a bunch of single task models, but it actually has like intermediate performance. I find this absolutely fascinating. I would love to talk, what your experience is and kind of what you think is going on here. Okay, so we were, and this is really kind of a challenge, and really calls a little like, oh, should I do single task? Like, what are my tasks? Should I group these cells together? Should I group those cells together? You have to really think about it. And we decided that we're going to solve it in this way that just kind of like brushes all of those concerns aside. And in this case, we're really leveraging the fact that we have this nice tree structure, right? So for all of these cells, there's some sort of parent that they came from, except for the very starting point, right? So I can actually reparameterize this prediction task as just trying to predict the differences. Now, I haven't actually changed the problem at all, right? Because I can always reconstruct what your value is by just walking down the tree and taking the differences that I have predicted. So the problem in some sense is identical, it's just like a linear transformation of the original problem, but it makes this kind of conceptual challenge of figuring out how to group cells or which things should be contrasted with each other really simple. I guess there's one simple solution. Okay, so we try to solve this problem, and everything that is predicted well is colored, and you can see it is actually just not very many things. It's kind of interesting because we're doing great on lineage, and this tells you kind of like that there are just specific contrasts that you can predict well. So all of these cell types, they're all different. The immunologists sorted them based on like their knowledge because they are functionally distinct, right? We don't still have a great way of understanding why they're distinct in terms of regulation. And so if you look at this diagram, most of the color edges fall onto major lineage transitions. Like, for example, we can predict B cells really well, but actually what we predict is this specific transition really well. We can't really tell the difference between these things. And a lot of these are actually just kind of activation. So generally we can predict activation fairly well. But we did have kind of one exception, which is the split going from double positive to CD4 and CD8. And these are CD4 and CD8 naive T cells. If you ever had to work with these cells, like doing single cell or something, they're quite similar, right? In their transcriptome and their epigenome. Really, like their main difference is whether or not they express CD4 or CD8. Like if you go to that locus, yes, you can tell what cell type it is. Anywhere else, it actually be kind of hard to know which kind of cell you're looking at. So, but we're doing the prediction we're getting is actually quite good. And then we can zoom on this. These are all the transitions that we could predict. So this is a transcription factor to now transition map. And so I'm just zooming in on this on the split. And one of the main things that we're getting is ZBTB7, which is also known as TH-POC. It is on the textbook map. And moreover, it is actually the sign, it has a negative sign, which is also correct, but is also what is known from the literature. And so that is something that we didn't actually get in the like lineage aggregated model. How am I doing on time? Okay. All right. So just a few things. I get a lot of questions like, why didn't you do this? Why didn't you do that? Sometimes like, well, we just didn't have time. But the answer to a lot of these questions is we tried and it didn't work. And what I mean that it didn't work. So we had some kind of metrics for success. And so one of the metrics is performance, right? We're always measuring performance, an internal metric, you can compute it. There are a couple other things we looked at. Stability across folds in the final layer, the final TF2 cell type layer. We wanted to get basically the same result over and over again, regardless of initialization. And we also had like recovery of cell time commitment TF. We had a fairly short list of things that we were like, you know, absolutely have to be there, otherwise the model is nonsense. One thing you notice is like, our model is really follows kind of the billboard structure, right? Because we have all these TF activations that just get aggregated and we lose all of our positional information. There's a big debate in the community whether we can have like, whether this is the right model or there's the grammar model, which things have to be like arranged in very specific orders. There's of course like very nice examples in literature where that is the case. Overall, it seems we've converged that like to the extent we have grammar is maybe it's weak grammar. But general, like any kind of like extra complexity we added, including China model grammar, we just had kind of failures. You can improve the performance, you can do complicated things, model performance will go up, and then you will lose the other two things. So just kind of like an overview where I think the challenges are here. And just to contrast, you know, deep learning with interpretable models. In some sense, like we still have a deep learning model in the sense that we're using PyTorch, right? Sometimes that's all people mean. Like I use PyTorch as deep learning, done. And we have layers, but a few things are different. So we have fewer parameters, that is true, but the model has to be, in contrast to deep learning, where you have like lots of paths to the correct solution, and that is literally why it works so well. In the interpretable space, you don't have that. You don't have that advantage at all. You're trying to find like this one correct solution. And I might actually, there's just lots and lots of ways to break the correctness of the solution while still giving like great performance. And so I guess just like I'm gonna show a little bit of ongoing work that's really preliminary. I am personally still quite interested in this idea of grammar. I know not everybody is. But what the solution that we came to is that like we just don't have enough data to model grammar. And this data set is not sufficient to model grammar. We either need more sequence or we need more outputs. More outputs is actually pretty easy because we have certain kinds of data where we have basic level resolution. So instead of predicting one value for the whole region, you literally get to predict like a whole profile, which has something to do with the biophysics of what is actually being occupied on the DNA. And so the base pair resolution is we have like a really famous model that's in this class is BPNet, right? This is kind of how the whole field started. This is the seminal paper that really demonstrated what you can do even if you have a black box model. Just to give like a little refresher. So BPNet is it operates on this like chip nexus type data, which is a really high resolution chip-seq. So you know, in the ideal case, like you get these chip-seq type offset peaks, but they're really concentrated like at almost like a TF footprint scale. right? So they're really centered. You can basically, in like a very clean experiment, pinpoint the exact binding site for the transcription factor. So in reality, the data kind of looks like this. It's a little messier in terms of like, you don't always get like really crisp, like symmetric peaks on both strands. So it can actually look kind of messy. And so the challenge, what BPNet did is that they just predict this profile at base pair resolution with a fairly complex, like it's the optimal layers of optimal architecture was a 9-layer dilated architecture. So lots of parameters. But then even though they had this black box model, they were able to do these cool things by just like moving things around in silico, right? So they were able to answer some questions about grammar by taking motifs and putting them at different positions with respect to one another. And so you can get these kinds of like activation maps that are dependent on distance. And one thing I'll point out here is that like we have some of these maps show a very nice periodicity. What's the periodicity? 10.5. It's the helical periodicity of DNA. And that makes a lot of sense. We get these things over and over again because certain things will interact better if they are oriented on the same side of the DNA. Sometimes. It's true for some things. So we decided, you know what, we're going to try to redo this model, but actually, instead of doing this thing in silico, we're going to get these parameters right out of the model. So that's what we've been working on. I'll just kind of briefly take you through it. So we have kind of like a base layer where we do like look at their DNA and we just find all of the binding sites for transcription factors. We're just working with the binding sites that we know matter for this problem. And then we have this influence kernel, which is like a large kind of up convolution or deconvolution kernel, which lets like a local site have an influence over something. So this kernel can make something that is binding here, make this site look all of a sudden more attractive to a different transcription factor. Okay, and so we have two models, which is like BPNet, which we re-implemented like 9 layers of convolution. We just followed what they did, but in PyTorch. And we have our kind of base model, which just has transcription factor binding sites, but no long-range interactions. And then we have the FFT for fast Fourier to do the long range that has interactions. And we managed to kind of definitely close the performance gap between the very complex model and the extremely simple model. And we look at the profile correlation, that's the left plot, and we also look at like across regions, how well you can do overall for a region. And for the profile correlation, we definitely see like a very nice boost from long range. And okay, I'm just gonna skip this. That's kind of, there's a part of this model that just outputs like the assay, but this is like in some sense like the nuisance parameter because you have binding and then you have the assay footprint, so you have to output like an assay kernel as well, and we can just show that what we get out of the model is very similar to what we got in the paper using like an algorithmic approach. So you do kind of learn it through optimization. But this is the fun part. This is the influence kernels. So they're still not as good as I would like. But here's one. It shows periodicity. These are a bunch of influence kernels for different motifs. And all of the ones you can look at the spectrum, and all of the ones that are yellow are the ones that show kind of an expected 10.5 periodicity. All of the ones that have green check marks are the ones that show that periodicity in the paper, in the original paper. So we agree, more or less. Right, so just to kind of wrap up, what should actually the goals of interpretable models be? So for me, I think this is, very beautiful. People have lots and lots of takes. So what I would like to have is kind of very factorized models, which have individual components that I can individually perturb, right? Turn off transcription factors, turn off the influence of one transcription factor or another and see what happens. And the other thing, kind of to contrast interpretable models with really free-form models, sequence function models, at the end of the day, they have to obey some kind of biophysical principles, right? Like if I have something that's interacting across a very long, that has very long range interactions, it has to be an actual physical thing, maybe a complicated sequence of events potentially, but it can't be magic. You can't have things interact like at long distances without actually some things coming in contact with one another. So I think if you kind of, we can try to build in some of these principles and in the process, we actually might take a hit on performance, and I'm personally okay with that. I don't know, maybe you're not. So, right, I just kind of want to acknowledge the people who worked on this. So, so for the TISO found paper, so Turl Balchi and working with Mark Abade led that work, and for the kind of the grammar model, I have a new student actually just joined the lab, so she did this all in rotation, and just a few months it's just been in my lab. I thought it was kind of impressive. So that's Jenny Hoop, and of course I'd like to thank the funding and collaborators. I'm happy to take questions.